Hello everybody, today we're going to talk about the sodium potassium pump, a widely used membrane protein complex that's utilized to establish membrane potential throughout our body. We wouldn't be able to have muscle contraction without this pump, so I wanted to go through it in a good amount of detail, and hopefully everybody will be able to follow me along. Okay, so first thing, this pump is a protein complex, like I mentioned, that is embedded in the phospholipid bilayer. It's a transmembrane protein complex, goes all the way through. Okay, and this again is the protein portion that's in blue here, and these are the phospholipids off to the side. So just to identify a couple anatomical structures here, there is a sodium binding site right here. Okay, this structure, and then these two are also sodium binding sites. There are potassium binding sites on the other side, and these are unique signatures here. They're not going to bind to just anything. They're only going to bind to, in this case, potassium, and in this case over here, sodium. Okay, and then over here we have an ATP binding site, and what's going to happen is ATP is going to come over and attach this. This has a lot to do with how this protein is made, very specific in terms of its conformation or orientation. So ATP is going to be able to attach to this location and only this location. When that occurs, it's going to activate a cascade of events. The first of which is that sodiums are going to enter this space and start to attach to the sodium bonding sites. So once these are full, what is going to happen at that point is that the ATP is going to release one of its phosphates or transfer a phosphate to this protein. This process is called phosphorylation. You can see the phosphate come off of the ATP. This now becomes ADP. But more importantly, this protein now has added a phosphate group. What this does to the protein is it serves to stimulate a conformation change. So phosphorylation, this term is widely used in biology, but that term, when you see it, you should be thinking an alteration to the shape of a protein. When the protein was phosphorylated, it's going to change shape. Okay, and that's the change in shape that we're going to see because of that phosphorylation. Okay, so the next step of the process is that ADP is going to be released, and as that occurs, these sodiums are going to be free to go out of the cell now. So the sodiums are moved from the internal environment to the outside of the cell. So outside of the cell, we're able to stack up sodiums. Okay, and now you can see this opening is open only to the outside of the cell. So the next step of the process is then going to be potassium ions are going to enter through this opening, and they're going to bind like so. Okay, and we are going to see the phosphate be released by this protein. Now, if you remember what just happened when we added the phosphate, the protein changed shape. So what do you think is going to happen if we release this phosphate? That's right. These phosphates, the release of this phosphate is going to turn this protein or change the protein back to its original conformation. Okay? And so now what's going to happen is these potassiums are going to be free to be unloaded on the inside of the environment, the inside of the cell, and we're going to be able to start this process all over again. Okay? So I hope everybody understands the steps of the process. That's pretty much the whole sodium potassium pump in its entirety. And the big picture here is that in doing this consistently, using energy, the ATP is energy, we have to utilize energy to get this done, but in doing this, we're able to establish a gradient. Okay, and the gradient can be thought of in a couple different ways. First, we have a bunch of sodium on the outside of the cell. We have a bunch of potassium on the inside, and I hope this picture does a good job at depicting that. But also very important is that we are bringing more positive charges to the outside. So every time we run one of these pumps, we bring 3 positive sodium charges to the outside of the cell, and we only bring 2 back across. So overall, if you're understanding this, the outside of the cell is going to be more positive than the inside. And that's what we consider to be a membrane potential, or a change in charge between the outside and the inside of the cell. What this means for nerve cells in particular is that because of this process, overall we're going to have a increase of charge on the outside, and there will be a lower charge on the inside. Okay, and what this equates to in nerve cells typically is around negative 70 millivolts, meaning that the inside is um, charged in a way that is less than the outside by a value of right around negative 70 millivolts, and that's only because of the sodium potassium pump. Okay, in a nerve cell, typically this would be the axon. You have leak channels, which are open all the time, that are going to allow sodium to come in, potassium to come out, and then we have these pumps, which are going to be moving these ions in both directions. Again, in order to ensure proper maintenance of these nerve cells, there has to be a negative 70 millivolt difference between the inside and outside of the cell. So here's what happens when a neuron fires. We got all these sodiums on the outside of the cell, and as soon as we open this voltage gated channel, these sodiums are going to just flood through here, okay? And that's going to alter my membrane potential, the charge differential between the outside and the inside of the cell. And a little bit down the road, what we're going to see is the same thing is going to happen with these voltage gated potassium channels. They're going to open, potassium's just going to flood across this membrane. Okay, and again, that's going to alter the charge difference between the inside and the outside of the cell. Okay, so when we're looking at a neurologic firing, what we're looking at here is these units called neurons. Neuron has dendrites over here, which receive information. It's gathered at this region called the axon hillock, where I have a ball representing the signal, and then that's going to travel down this very long, thin axon. Sometimes these things can be up to a meter in length. That's how long they could be. But it's going to travel all the way down here, and then down here at the end we're going to have these vesicles that are going to release neurotransmitters out into a junction here, where it's going to meet up with another cell, a target cell, and that's the gist of how a neuron functions. But it all comes down to the functionality of these sodium potassium pumps. So we're going to innervate this neuron, and you can see the signal is traveling down the axon all the way to the end of these synaptic bulbs. And what's happening here is that along the way, these red lines are representing these sodium potassium pumps, and every time we pass one of these with the signal, because of the alteration to voltage, all comes down to that voltage gated channel. Once we change the voltage in the interior of the cell, that channel opens up, and once that happens, of course, all the water that we have stored behind the dam, in this case sodium ions that are outside of the cell, are going to come rushing in. When they do, they're going to alter the environment of the inside of the cell in a way that adjacent sodium ion channels are going to open up. These are, again, voltage gated channels, so because the voltage is altered in the surrounding area, these things open up, new sodium ions flood in, and in that way we move down the neuron, and we can cause neurologic firing in the brain or contraction of the muscle or whatever lies there at the finish line. Thank you for listening. Have a good day.